and welcome to the fourth of our 2021 virtual event series. My name is Kelly Moran, and we are just launching today our 2021 expedition season. For the next 6 months, these events will be opportunities to hear about the excitement of all of our expeditions, preview the stories coming to you from sea, meet and engage with the various team members on board the Ocean Exploration Trust and our Corps of Exploration, and talk about science, technology, engineering, and the history that excite each of our team members. We're glad to have you joining through Facebook and also YouTube. If you have any questions or comments throughout this program, feel free to type them in the chat, and we'll try to get to as many questions as we can throughout this program. We'd also love to hear where you're coming from, so feel free to type in where you are today so we can give a shout out to your hometown or the different countries that are joining us. Today is the very first day of our expedition season, and we're super excited. We plan to launch the ROVs this evening around 6:30 p.m. Eastern, 3:30 p.m. Pacific, so stay tuned to that on our website, nautiluslive.org. This will be our first live dive of the season, and we have invited the researchers who are on board the ship and also an artist who's participated and done some of the science, but in art involving what we're going to be talking about today. So when we talk about the deep ocean, we also, we really frame things in really big scales, super deep, where we explore with the robots, really bright lights on the ROV Hercules, really big machinery, really large ships like EV Nautilus, and also just how big our audience usually is during these dives. But something we don't always talk about are the really, really tiny things we might be studying and exploring in the deep sea. And that's what we're going to talk about today. These things are called forams, and we'll get to a bit more about what exactly they are in this presentation. So first, I want to bring you on board EV Nautilus and talk with Dr. Joan Bernhard, who is on board the ship. Hi, Joan.
Hi, nice to see you.
Great, you too. So first question, it's the first day at sea. How's it going? How's being on EV Nautilus?
It is beautiful weather. It's almost flat calm, so it couldn't be better. And so that's wonderful. It's sunny. The food has been great, of course, so that's always good. It's an awesome team. I've never sailed with Nautilus before, but everyone is really enthusiastic about their roles. And so I'm really looking forward to getting the Hercules down on the seafloor later today.
Awesome, great. I'm really glad to have you on board for the first time. It's great that you're out there. Let's jump right in and talk a bit more about the topic of this presentation, forams. What are they?
Right. Yeah. So first of all, they're actually called foraminifera is the proper term, but it's such a mouthful that we just try to call them forams for short. It's much easier. So they're a kind of amoeba-like protist, and they occur in almost all ocean habitats. So they're a single-celled eukaryote, if you know that terminology. They have many, many different shapes. They come in many different sizes, and they can be, so here's an example of some of the shapes. These are actually forams from Santa Barbara Basin. They also come in a number of different sizes, from just a few microns all the way up to actually centimeters, like the size of a tennis ball. And so some of them make their shells out of calcium carbonate, more or less like a clam shell. And so those shown there, those would be calcium carbonate ones. But there are other ones that take sediment grains and glue them together with an organic cement. And then there's other ones that actually don't have any kind of hard shell at all. It's just an organic covering. And some of those forams that take particles and glue them together, they can be highly selective. And yeah, they're really interesting. So forams have pseudopods. It's a kind of specialized pseudopod or false foot. Oh, so here we have some examples of a calcareous foram. And then on the right is an agglutinated foram. And then the one in the blue image, that's one of these, quote, soft-shelled forams that just has an organic covering. So we can start talking about the pseudopods now. So they're actually called reticulopods. And so those are false feet. And the thing that's special about the forams is that they have adapted so that they actually branch and then come back together. So they actually make more like a network. And this allows the forams to be very, very, their pseudopods or reticulopods are very strong. And they use those to capture prey and grab food particles. They also use them for moving around. And so this is what has made them really, really successful in the ocean.
So we'll talk a bit about, oh, sorry, Jen.
No worries. Go ahead.
Awesome. I was going to ask, we'll talk a little bit about this location in a second, but knowing all of this and how, they can make new ones and how they're so important, can you talk a bit about what role do they play in understanding more about our ocean, or, why are they so important to really studying the ocean and the ocean as a whole?
Right. First of all, it's important to understand that they have been around since the Precambrian. Most likely, we don't have any fossil evidence for that, or shall we say, people argue a little bit about that in the scientific literature, but I believe that they evolved before the Cambrian, so before the animals evolved. And so the first ones had these organic coverings, and they are not going to fossilize unless you have very special circumstances. These images are a number of specimens of one species from Santa Barbara Basin, and this is one of the ones we're hoping to capture later today and study in more depth. But so these evolved a very, very long time ago, way, way before the dinosaurs. And so over time, these the original species, of course, went extinct, and then, of course, other ones evolved. And they're very diverse, and they've had a very long fossil record. And so the foram fossil record has been used in the past to interpret both the age of a rock or a geological deposit. And you can see an example here of all the variety that you could get just in one sample. So not only can you age date a rock, but you can also determine what kind of environment that was when the rock was formed. And so then you can use that information, and that has been used for hundreds of years in the exploration of for oil and gas. So they've been used a lot in the petrochemical exploitation, if you will. But they're also really, really important more in the last 60 or 80 years, because we can analyze the chemistry of the calcium carbonate shells that they make, and they make that carbonate under certain environmental conditions, and they record the environment when they grew. And so those are used to interpret past oceanic conditions, which is also helpful to understand past climate conditions. So using the chemistry in the foram shells it's just absolutely foundational to the study of paleoclimate, which is really important for understanding our climate today, as well as predicting future climate scenarios. So it's really important in the climate change field, shall we say.
Awesome. And then I know the ship just, is in Santa Barbara now, or the Santa Barbara Basin. What is the significance of this location, and why are you there studying forams in this spot?
Right. So So let me just say, so I started sampling Santa Barbara when I was a PhD student, and that was back in the 1980s. So this is a very long time ago. And so I've studied it quite often just using over-the-side methods like a box core. So, and this is will be the first time that I've ever used an ROV in the Santa Barbara Basin. And so the reason that the basin is so interesting and why I keep coming back is because it's rather unusual because the seafloor is often stagnant, if you will. So so the oxygen concentrations get very low because the water doesn't circulate much. And this has to do with a variety of factors, including the geography of the seafloor, the layering of the ocean, and then also the chemistry. So often you have a microbial mat, very well-developed microbial mat spanning most of the bottom of the seafloor. And this is an image here showing the bottom of Santa Barbara Basin, I believe, in about 10 years ago. And so that white material is actually bacterium. It's a colonial filamentous bacterium. And anyway, so this will be the first time that I've used an ROV. And the reason that we're using the ROV is because we can do different things than doing over-the-sides methods. So Santa Barbara is more or less a natural lab used to understand what will happen in the future when the oxygen is decreased in the ocean. And that is also happening with climate change. We're getting much less oxygen dissolved in the ocean in certain places, and those areas are expanding. So that's impacting the ecology. So, so in Santa Barbara, we really don't have very many animals living there, but we have forams that do unbelievably well, like 10 to 100 times more abundant in Santa Barbara than outside the basin. So, and the species that really thrive in the stinkiest part of the basin, if you will, so it really can smell like rotten eggs. They have a very different cell structure. So they're not all the same. So these different species have gone about these living there in different ways, and that's what we're studying. And one of them is just really, really fascinating because it takes chloroplasts, which are an organelle that algae and plants have, and that's what captures sunlight to make their energy. But this foram is doing this, and they're just full of these chloroplasts, and they steal them from diatoms. So we call this kleptoplasty. And it's really interesting because these are living deeper than 500 meters water depth, and generally speaking, you don't have photosynthesis deeper than, say, at the most 200, maybe 250 meters in a really clear ocean, not near land. So that's why we're really interested in this site.
That's awesome. Thank you. I'd love if you could pass the mic over to your colleague next to you, and we can introduce our next speaker. Hello.
Hi.
Awesome. So, Chris Powers, what are you doing on board the ship? What are you studying, and why are you interested in forams as well?
So I'm here helping out Joan with the collection of these forams. And my interests in them are kind of centered on their metabolism. So Joan mentioned that these guys are found all over the place, right? And in addition to be found all over the world, they're also found in really diverse habitats too. So you can really find them in places, really deep in the sea where there's no oxygen, places where there's lots of really toxic compounds, and we don't really fully understand how they're actually doing that at this point. So my goal is to try to get a handle on the metabolism and try to figure out, what are they eating down there? How are they processing it? How are they using that to grow? How are they producing energy? Things like that. So here is kind of an analogy that I built to try to communicate what metabolism is, right? So in humans, everyone, eats pretty much the same stuff, but some people have different nutritional needs and metabolic needs. And some people are gluten intolerant, some people are lactose intolerant, and these, require different metabolisms. If you eat something that's bad for you, you kind of get sick from it. And some forams are the same thing. And on the right, I have, the analogy of forams, and I use the example of oxygen because we have forams that are abundant in no oxygen. Forams that are abundant in, when they have really low oxygen, it would typically be pretty bad, but, there's whiffs there, as they say, and some of them are abundant in abundant oxygen.
So basically, oxygen is how they form different shapes or how they get their different unique kind of structures?
So that would be how you would typically think of something like that's how we get our energy, right, is by utilizing oxygen. And the remarkable thing about these forams is they actually, many of them are so abundant, 10 to 100 times more abundant in these sediments that don't have any oxygen. So there's a question of how they're doing that. And for example, one of the hypotheses is there's lots of nitrates down there using nitrogen instead of oxygen to make energy.
So how on the ship now and first dive later on this evening, kind of what's the plan for that? How do you expect to be taking these samples and what are you going to do with them once you bring them back on Nautilus?
So we're going to go down and collect sediment using a push core. So it's going to go down with a big tubing of plastic, basically, right? And we sink it all the way down in the mud, and we will bring it back to the surface, and we'll have this nice core of sediment, and we'll be able to sample the top, and the forams will be in there. Yep, just like that, it's going to stick down in the bottom, and then when we pull it up, the forams will be in the top layer of the sediment. And Joan will use some microscopy to pick them all out through the mud, isolate single cells, and then some of them are going to be treated on board. Some of them are going to be brought back for all kinds of fun experiments. So, yeah.
Awesome. And how many forams do you think would be in one push core?
Yeah. How many forams would be in about a push core? Ooh.
Wow. If conditions are good, we can have 10,000.
If conditions are good, we can have up to 10,000 forams in a single push core. So they're really, really dense in those living ones. Yeah, they're really dense down there. One thing that we are going to be doing with the push cores that I didn't mention is we're going to be leaving some of them down there for a little while, doing some incubations down at the sea floor. And some fixations down there, kind of because, we have these light absorbing processes going on, and we're trying to see if we can stabilize them before they get exposed to any light. So we don't want to bring them up to the surface and potentially expose them to light. We want to get them while we're down there, which is pretty exciting.
That is exciting. And I know, you're all from different locations and somewhere near each other, University of Rhode Island, Woods Hole, but what's going to happen to the various samples that you're taking over the next few days? Do you get to bring some back for your program, or are they all going to one specific location?
So many of them are going to be incubated at the seafloor and then collected on ship and stabilized on ship. And then there's going to be mud samples that are going back with Joan. And then I'm actually going to be working with her in the next week, processing those samples back at Woods Hole. So that's not going to be happening at URI. That'll be at Woods Hole.
Awesome. Well, we're very excited for the upcoming dives. I'll come back to the ship in a second, but I do want to introduce our third presenter today. We have Terry Conrad, who's an artist who has worked with Dr. Bernhard on these various forams and designing artwork around them. So I would love to bring you in and have you introduce yourself.
Great. Thank you.
Awesome, Terry. So can you talk a bit about, how you got into this and how you wanted to create art around forams and work with Dr. Bernhard, in this type of environment?
Sure. I think I should first say that I met a scientist named Samuel Bowser, who actually, Dr. Samuel Bowser, Dr. Joan Bernhard worked with Sam for years, and he actually helped make the connection. He saw my work in a show, and he thought it would be good that we had a conversation, and some collaboration started to percolate. And then eventually I got to go to sort of shadow Joan and a crew on the Robert Gordon Sprawl going out to the Santa Barbara Basin, actually. And that was in 2018. And here, I love the fashion of what goes on in the trip on the ship. It's and I have a little bit of artwork actually that kind of references that later. But I knew so little about the science, and Dr. Bernhard was so patiently and continues to patiently teach me about these organisms and some what's at sea in general. So I was able to make quite a few projects, whether they were sort of studio-based projects, like the image over my shoulder, which is an image of Bolivina pacifica, which is, I believe, a foram that they might be collected out at sea soon on the Nautilus. Dr. Bernhard can let me know if I'm wrong there. But so I made quite a bit of studio-based work, and then also sort of performative-based work as well. So this is an example of two of my etchings. I am a printmaker and I teach printmaking. I'm very much interested in the idea of experimental printmaking. These are prints that actually, in both cases, use mud from the Santa Barbara Basin. So use some of the mud that we were using in those box cores, as well as the push cores that we were just mentioning. The image on the right there was actually an embossed image, and the ink kind of sets in there. But the image behind me here, this is more traditional inks mixed with the Santa Barbara Basin inks that I've been calling them. I've been calling them Santa Barbara Basin inks, but they're actually mud that was generated. I actually packed my suitcase with a bunch of mud when flying back before flying back to New York to my studio, and actually was able to use this mud. I'm still using my collection of mud to make a variety of inks.
That's awesome. And how long does your previous samples of mud and sediment, how long do you think you'll have until you run out?
Oh, I need to get some more soon, actually. So hint, hint, if anybody has some that they want to bring back to me, that's really great. I was really on the voyage, it was really sort of, everything was totally new to me. I started to see the connection of why Dr. Bowser thought that this would be a good opportunity for me in seeing how the research was conducted, the tools used, even the way the processing of the mud in basically this walk-in cooler where all the forams and seawater are sort of put into different containers, and then they were sent back, in most cases, to Woods Hole Oceanographic Institution.
That's awesome. And some of the, work that you're continuing to do, do you have plans to keep creating more artwork? I know you're running out of the sediment from your previous expedition, but do you have plans to continue making more foram art, or it was kind of a, smaller portion of your art?
Oh, I actually, Dr. Bernhard and I are talking about more projects. I'm really interested in, like I mentioned, the tools that are involved in the scientific research, as well as the organisms. And so the goal, yes, is to make more kind of more work ruminating on the foraminifera biology and habitat.
Great. And it's kind of an unusual topic for someone to start creating art around, but what really drew you to this, besides meeting the individuals that you've met, but what kind of captivates you to keep creating this art around not only the sediment and the mud from various locations, especially Santa Barbara Basin, but just forams in general? What about them, do you really want to capture as you're creating?
Well, interestingly, as a printmaker or thinking about experimental printmaking, there's something that Dr. Bernhard mentioned earlier, which was in reference to the calcium carbonate shells, how a foraminifera is a record of the environment over time. And I like that idea of trace, thinking about time and trace. And that's, thinking about drawing or mark making, that's what happens in printmaking. And I'm just kind of fascinated by the ways in which foraminifera are adapting. It was mentioned earlier about kleptoplasticity, and I really enjoyed the chart that was just pulled up earlier, the metabolism chart. That's fascinating, how different levels of oxygen were actually changing the form or the shape, the visual look. And so, yeah, I feel very lucky to be able to be inspired by and learn from scientists. And I do feel that my work is, I'm interested in that, not just sort of, making artwork that goes in a gallery, but the whole connection, the process and experience that can be involved in making. The image up here is actually, in a previous life, I worked as a costume and prop technician on Broadway. And so I was invited to the University of Michigan, landlocked University of Michigan, and did a project. And I was invited by a class that was an experimental fashion class. And so we basically made a bunch of foraminifera costumes. And everything was made out of found materials. That's sort of something I do in the work. And so it was basically, we made this presentation kind of mimicking what was going on the ship, on the Robert Gordon Sprawl. And so there were chemists, and there was a lead scientist, and there were all these characters. It was we took some artistic liberties, and the forams were different colors as well as large human scale. And so then we were also making prints with mud on the sidewalk of Naniella Stella, which is a foram that was kind of marked throughout the sidewalks. And really what it was out in the public, and it was an engagement, this effort was really to start a conversation about what even foraminifera were.
Those are awesome. And I feel like we should have shipped them out to Nautilus for this expedition and wore them out on the back deck or something. Awesome. Thanks so much, Terry. I'd love to pull you back in if we get any questions from our audience, but I'd like to hop back over to the ship for a question from our audience right now. And they're asking, how do the mud samples stay fresh after being collected and put into the wet lab? So kind of what's your process, either Joan or Chris, whoever wants to answer this one, but kind of what's the process after you collect the samples of bringing it back into the wet lab and, maintaining the forams and also the mud samples for the next week or so?
Well, some of them we're preserving them on the seafloor. Some are going to be immediately preserved, if you will, on the ship. And then I have to actually pick the forams out immediately. So I'll be doing a lot of microscopy on the ship. Hopefully we get the species that are what we're looking for. And then some we take back live. I've taken them back live since the mid-90s. So we have a way to secure them in high density polyethylene bottles. We put seawater over them and no airspace at all. And those bottles can stay, as long as they're cold, they can stay fine and keep the same species living, including ciliates and flagellates and the bacteria. for months, not years or anything. But as soon as you open the bottle and oxygen gets in, then that's too much oxygen in some cases, and then the community will change. So we can get them back to the lab to do things over the next month or so. After that, we don't generally use the live ones for very long.
Thank you. Yeah, and we're getting a lot of questions about the wet lab and everyone interested in kind of who's on board right now to help with this process. So there is one question from YouTube asking, is there a camera in the wet lab? And the answer is yes. So we can live stream our wet lab so everyone can kind of see the process and see everyone working in there. But a question for you both is, can you talk a bit about the other scientists or team members who are on board the ship right now? Is everyone studying forams, or is it a mixed group of scientists helping in this process for the next few days?
So we have, there's a team, well, there's a graduate student from Harvard who is part of Pete Gergis's lab, and she is in charge of deploying what I would call an elevator, or it's an instrument that will stay down on the seafloor and do some analyses, over time, take water chemistry samples, shall we say. And we will have a bio box on that. So we're going to do an experiment on that. And so that'll come up in September or October or something like that. And then we also have a graduate student from UCLA. And so he's in a lab that works on bacterial processes. And so both he and the woman from Harvard, Brooke, they will be collecting cores and taking the entire core back to the labs, their labs, and doing analyses on those. And then we have an undergraduate named Sarah Lott from Wagner College, and she's out here to do some microelectrode profiling, so analyzing the chemistry of the sediments that we'll be studying on very fine scales. And then my co-PI on this project, one of them is Ginny Edgecombe. She's out here with me also. So she, since we got rid of quarantine, she could come. And so I believe that's all of the scientists that we have out here this time.
I think that's one of the best parts of, EV Nautilus and exploring in general, is that you can have all these different types of students or PhD levels or experts in fields all studying different things, but a part of one team for one expedition, which is pretty exciting that there's a whole bunch of people out there right now studying roughly the same, but all different aspects of that area. We did have a question from YouTube come in asking, is maintaining pressure levels of the samples essential in these push core samples for the forams? Do we need to maintain any pressure levels?
We're only working at depths at the most 600 meters, and that is not really a problem. It's pressure starts to be a problem. Well, it depends who you talk to, but definitely you have to really kind of worry about it deeper than about 2,000 meters. Some people might argue around 1,000 meters, but we're at 600 and it's not a problem for these. They're, again, single-celled organism, and really their issue more is the oxygen. They don't like it when there's too much oxygen, and also, of course, temperature.
Great.
Salinity, but that doesn't change so much. Yeah.
Someone's also asking, is there a team on board, or is it someone's role to take field notes, observations about what you're seeing, where you are, things like that? Is there someone on board who's taking notes, or is that you?
Oh, I don't really have time to take notes. I try. But no, I mean, there's the whole Nautilus team. That's the great thing about working with Nautilus, is that there are people that it's their task to do the note taking of when samples are taken and what was going on at the time. And then, of course, we have we also have chemistry being monitored over time, and so we have a timestamp for that, so we can figure out exactly what the oxygen was at that point. The bottom of the basin is pretty flat, so it's not like hydrocarbon seeps or hydrothermal vents where you move a meter and it's totally different. So it's not a habitat at all like that, which are more point source type of thing. This is rather monotonous, you might say, but on a vertical sense, things change markedly within the top 2 millimeters. Generally speaking, in the sediments. So they look kind of boring from a plan view, map view, but it's the small scale vertical sediment chemistry that varies quite a bit.
Great, thank you. And I have one more question from the audience, but for anyone who's joining in, who's interested in life at sea or doing what you do, or just fieldwork in general while at sea on a ship, what are you most looking forward to over the next few days? And what's your favorite part about being on an exploration vessel?
My favorite part will be at the end when we've gotten absolutely every sample that we could possibly want, and they were all perfect and everything worked fine and no one was hurt. We didn't lose any gear. That would be my favorite part of the cruise. But I also enjoy the sunsets and also watching the dolphins. And we've already had a nice big pod of dolphins go by this morning. I was out, I saw one, and there could be more. I don't know. So that's always fun. I also really like to see flying fish. Those are always really fun. So.
Oh, God, that's a great view of the back deck right now and you guys steaming ahead. So I want to thank both of you for joining us. Terry, we've also had some viewers in the audience who are really loving all of those costumes and looking at the forums in life form over different people. So I'm so glad you showed us that today. Thank you for joining us today as well. For anyone that's joining us who is interested in life at sea, or artwork about life at sea, I highly recommend, especially for EV Nautilus, going on our website and looking up our Science Communication Fellowship and also our internship program for college and recent graduates. It's a great opportunity to possibly get on the EV Nautilus and work with the scientists who are on board in various expeditions. So I do recommend checking out nautiluslive.org/education for any opportunity to come out on the ship or just learn about the different folks who are part of our Corps of Exploration in all aspects of being at sea. So please check out our website. I want to thank all of you for joining us today. I really learned a lot about forams and why they're important, and just, the excitement of this very first expedition for our season. So thank you, Joan, Chris, and Terry, for joining us today. And this series will continue throughout the entire 2021 exploration season with expeditions on EV Nautilus, again, launching today off the coast of California, but we will be bringing you adventures from the East and Central Pacific. So we will be going until the end of December. It's a pretty long season this year. Please share this channel with all of your friends and just keep exploring with us at nautiluslive.org. Again, our first dive will be this evening, so tune in. We are streaming 24/7. You can see what we're doing at all aspects of the day and night, whether we're mapping or steaming ahead like we are now, or diving with the ROVs, which will again be about 3:30 p.m. Pacific time. And join in for our next live event, which is happening on Monday, July 19th at 12:30 p.m. Pacific to get a preview for our next expedition. So thank you all again for joining us, and I hope you tune in to nautiluslive.org for our very first dive this evening. Thank you.